Okay, so hello everyone. We've got Emmanuel Martini here. He can introduce himself, but he's also part of ABIC. And the course we've been going through in the last couple of days, we set up together. So some of the slides that you've seen and some of the examples that we've been doing, he was the one that created some of that. So yeah, and you will now do this last session. But yeah, you can introduce yourself and then go from there.
Okay, let me start. Okay, so good afternoon. Here is Emanuele Martini. I am part of the African Bioimaging Consortium. Moreover, because I live in Uganda 8-9 months a year. At the moment, I'm in Italy. For the last few days, I will be back in Uganda on Sunday. And I'm a part of being a ABIC member. I'm a researcher in the University of Milano and in a, let's say, oncological research center where we study, moreover, cell migration. And so today we will give, I will give you a talk about how you can study cell migration. It will be, of course, an introduction, but I think you will see most of the main topics and main stuff you can do to study cell migration. So what you will see is, as I said, a brief introduction to cell migration. Then we will try to analyze a classical cell migration experiment. We will try to do it with an hands-on. So we will try to do all together and see how far we can go. And then I will just give you a very quick introduction to script automation with Fiji. Very quick, because we don't have so much time, but it's just to let you know how you can do a script with Fiji very fast. Okay, let's move on. So why cell migration? Cell migration plays, of course, a central role in the development and maintenance of multicellular organisms. It's important for embryogenesis, tissue formation, wound healing, immune response, and of course also in cancer is really important because it's one of the processes that leads to invasion and metastasis in cancer dissemination. So this is why it's important. What is basically cell migration is exactly check how a cell or a group of cells moves in certain kind of condition and where they go, how fast, and something like that. Okay. So in the first part of these few slides, I will show you what is tracking. So tracking, it means, okay. So tracking is basically following particles over time and describes trajectories. So it has two-step process, as you can imagine. There is one step is where you detect and find the object. So in our case, can be cells, but in outside of the world of biology can be the, I don't know, when you see CCTV camera and you want to follow people or cars, you have to detect the people, the cars. So your object of interest in our case will be most of the time cells and nuclei. Or you can, and then the second step is the linking, no? Frame by frame, 1 minute to another minute, understand where an object is going and link it in different frames, to understand which is the same object in the frame 1 and connect it to the same object in the second frame and in the third frame to be able to describe the trajectory. So just a very, very theoretical introduction to it for bioimaging. Bioimaging specificity in tracking. There are good things and bad things, respect to, I don't know, following a car or something like that. The trajectories, one bad things when you deal most of the time with biological cells and particles is that the trajectories can be erratic and complex. So they can change direction in an unexpected way. They can move faster than slower. So it's a little bit not so always the same movement, so you have to consider it. Then a lot of time, as you can see in the next experiment, in the next movie I will show you, there is a high density of particles. So you have a very large number of cells, of nuclei that you want to follow. This is not always true outside of the biology. If you think, as I was saying, to follow one person entering as a thief in a house, it's just one and you want to follow him, something like that. And then a good thing instead is that you can do it offline. What does it mean offline? And most of the time you do it offline. You acquire your experiment, your time lapse, your movie of cells moving, and then you can analyze them in another moment. You don't have to do it in the same time when they move, when the cells move. It's you can imagine it as a very different case respect when you are in a war and they launch you a missile or a bomb and you want to track it. You cannot track it offline because the bomb will jump on your head. No? Instead, with cells, you can acquire your experiment. You don't have to take care too much about computational power. It can take 3 hours, 4 hours, 5 hours. You can just wait. You can do when you want. You do the experiment, then you track it. Let's be a little bit more precise in the two steps I said before, the particles detection and the particle tracking and linking. So the detection, I want just to stress one thing about it, that is not always necessary segmentation. I think in the other lectures and the other talks, Rensu introduced you to the segmentation theory, and then what it means, just to be sure that everybody's on the same page, segmentation means be able to find the real boundaries, the real outlines of the object. This is sometimes needed in tracking detection, but it's not always needed. A lot of time you just need to identify more or less where is your cell, more or less where is your nuclei, but you don't need to find the real boundaries of the cell or of the nuclei. It really depends. Sometimes you are also interested in that, and so you have also to have a very good part of segmentation. It depends with the biological question of your experiment and of your project, but it's nice to know that you don't need always segmentation of your particles. Now, also for the linking part of the tracking, I just want to introduce very few basic theoretical consideration. So most of the time when you want to link, so to identify the same object frame after frame, you can have two approaches. One is heuristic, so it means you just you put some parameters like distance between detection, intensities of detection, if you have the boundaries, the area of the detection, something like that. And then you have something more probabilistic, or let's say modelistic, mathematical models. So you can put some model assumption. So if you expect a random motion, so they go by chance, or if you expect that they always go straight, moreover straight, or if they have a constant velocity or a constant acceleration. This part goes inside some algorithms of linking, and they solve the problem on reducing the cost of linking the particles that you have identified in the step before. What you obtain is something like that. This is a time-lapse experiment when each object you see is a nuclei tagged with DAPI, with a fluorophore. And then what you are really seeing is like the detection part where they found, when the algorithm find the nuclei. But as you can see, it's not really finding the boundaries of the object. It's not really doing a segmentation. It's just identifying the area where the object is, and then you can see the line following. That is the linking step we saw, to identify and follow the particles and draw the trajectories. And then from there, what you can do is extracting different parameters like the velocity, the directionality, if they move randomly or not, and then you can do your consideration about your biological project. In Fiji, you have really many tracking plugins. So I would just want to introduce the fact that they are available. I don't want to go too much inside and see how they work. The most used one are the particle tracker, but moreover the TrackMate. And I would just want to show you that I said before, there is some particle detection morphological parameters, like the radius of the object you want to find an idea of how big is your object. And then, for example, here you can have the dynamics that you can expect, if random or it has a or you expect a constant velocity. So here you are giving some constraint to the algorithm to be able to find better the same particles in each frame and link them well. Then you can see some other parameters that always are present in the tracking algorithms, that are the displacement. So let's say the longest distance you expect a particle to move in one frame. So it's like telling to the algorithm to don't search particles at a distance of more than 10 pixels or 10 microns. It depends on the calibration. And then there is the link range, that it means that even if a particle is not found in some frames, for how many frames you can search the same object, the same cell, the same nucleus. So in this case, you are telling to the algorithm that you can find the same, to search the same particles for 2 frames. This is why, because sometimes moreover in biology, the particle can be out of focus, can go to another plane and then reappear. So you want to give to the algorithm some flexibility to search not only in the frame after, but also in maybe 2 frames after, 3 frames after. And then you have, of course, some algorithm. I'm not going to say the details of that, but you have different, more elaborate, more sophisticated tracking algorithms to link the particles. Then one important thing to know is that, as in other, let's say, biological experiment and analysis, sometimes it depends on the complexity of your experiment. You prefer to have a semi-automatic or manual way to analyze your experiment, and also for tracking is present the opportunity to do manual tracking, in the sense that you follow by yourself instead of following all the cell by yourself. It helps you to follow by hand, so clicking on different frames a cell, and then of course it's better to have something like this, because where is it? It's better to have something like this, and then if you have nuclei with DAPI or tagged some way, it will be quite easy to find algorithms able in automatic to follow all of them. But if you have something like the first one I show you, like this one, a case like that, when this is just a bright field phase contrast acquisition, so you didn't tag any nucleus, you just have the image of the cell. Finding an algorithm able to follow and find this kind of object that change a lot the movement, that change a lot its morphology, it's not so easy. So maybe at the beginning, and for some, I don't know, time, you do it by hand, because it will take maybe longer to find a very good algorithm able to track all of them than do by hand, I don't know, 50 cells for each condition. Okay. And another thing, I will go back to the other slide, to this guide. What you can also see is that sometimes these algorithms are even more sophisticated. In the case of TrackMate, it allows you to also follow the phylogenesis of the cells, in the sense that you will be able to follow the mother and the daughters and see the daughters, how they move, and then so also follow the duplication, the doubling of the cells. It's interested on it or not, but it's very tracking is a very developed field in bioimage analysis, so you find really a lot of nice stuff around. Okay, so what can you extract usually for this kind of experiment? You can extract the velocity, the length of tracks, so for how long for how many frames you will be able to follow that cell, the persistency, persistency is for how long, for example, it keeps more or less the same direction, or it goes always, or it's moving more randomly. You can find directionality, distance. I mean, there are a lot of parameters that you can take out from this single cell tracking and trajectories. Usually then you can put something, you can plot, sorry, something like that, that is, each trajectory is plotted. Here, I don't know, it was an experiment. The red one was a kind of condition with some treatment, and the black one was with another condition. With a plot like this, you can already see, without doing statistical or parameters extraction, that, for example, the red condition was moving much less respective to the black condition. This is like what you usually can do about tracking at the single cell level, so here at the single particle level. If you have any question, you stop me, Renzo, you tell me, and then because I don't see the audience, so I don't see if someone is raising hands. So if you have any question, just
you can
speak.
carry on. Everybody's still happy. Thanks.
Oh, okay. Now, in the next longer part, we will explore something different, something related, but a little bit different, that is the so-called collective cell migration. So it's a complement this field of cell tracking, but you want to study, instead of only one cell per one cell, you want to find parameters and way to study cohesive groups of cell moving. So you want to see if the cells correlate their own movement with the neighbors or not. So this is, as usual, you can see it always in the field of the importance in cancer research, but not only in cancer research, but in cancer is one of the complementary strategies to promote cell invasion and metastasis. So you can see single cell movement is a way, when they are all together, they move differently. This is, for example, an example of one of our papers. What you can see is that the cells are always all of the same condition, but they are plated at different density. And what you can see is that at different density, they move differently. Okay, here is just a very nice plot of the situation. You have a control and a treated. The control doesn't change in our experiment too much the velocity based on the density, but if you see the treatment, if you increase the density, they really move much faster. And then in the next slides, we will try to see how we can calculate and extract this kind of parameters. But here is very important to understand this part here. So when they are alone, they can have a single cell movement. When they are together, it's not always that they go at the same way they were going when they were in a lower density. They can really have a very different dynamic behavior. Okay, let me drink 1 second. So, to study this kind of collective movement, there are mainly two ways, okay? Two kind of experiment. One experiment is the so-called directional movement. So, the starting point is the same of this one, but what you do is like, let me go back from the beginning and move it by myself. So you go yourself like in this one on the right, and then with a tip, you do a scratch and you create a wound. And then you want to see how your cells close the wound, no? And then how speed they move, and this kind of information. Okay, this is a directional experiment. It's a directional experiment, of course, because you are some way imposing to the cells a favorite direction, that in this case is the vertical one, because they have space in front of them, no? And then there is another one that is the confined movement, and it's usually referred to cell streaming experiment, and the other one is usually referred to as a wounding experiment. You grow the cells at a certain density, in a certain way, and then you just let them go and see how they move. So here you are not imposing a direction to the movement, you just want to see how they move in a situation like that. In the next slides, we will explore in a theoretical way this kind of experiment, because it's a little bit harder to do an hands-on in 1 hour. And then instead we will try to do an analysis of this one by hand. So we will try to do a Fiji analysis of this in 40 minutes, in 30-40 minutes. Okay, so I already said, but we are interested in the movement and the correlation and in the way they move as a multiple cell, less interested in that, how they move at the level of single cell. Okay, so I already say that we want to see velocity, directionality, coordination of the cells. There are many tools to do it. One of the main and most used one in this kind of experiment is the so-called PIV, or particle image velocimetry. This is an algorithm, a tool, a strategy that has been developed since a lot of years outside from the biological systems. It was more related to physical system, to see how a fluid, how I don't know, water moves in a confined space, how they move the fluidity of some liquid or fluid. Then someone thought that it could be also useful to study this kind of experiment, because at the end of the day, also this one is like a fluid moving and so full of particles, so maybe it will have worked, and indeed it worked well, and then now is quite really used a lot in the field of cell migration. I will try to explain it a little bit in a little bit of the details, but the main part is something like that. You know, you have in your hand a time lapse, so you have different frames, okay? And what you want to do is comparing frame by frame, no? So 2 frames at a time, a couple of frames at a time. You identify a grid, so you do a grid, in this case 9 squares, but it's just an example. And then you want to see in each square, where is the movement, so where is the movement of inside this little square of the intensity present inside the other square at the other frame, okay? So you want to compare this square with this square in a couple of frames, in frame 1 and frame 2. What you do is you don't want to recognize objects. You are no more doing nuclei identification, particle identification. You are only relying on the gray level of your image. And what you can do is find a mathematical function that find similarity between this square and this square, where there is more similarity. Okay, and then if you do it for all the squares for the frames, what you are going to do is to have a velocity field, or a vector map, or a flow field. It depends on how you want to call it. But what it is like each arrow is the estimate of the movement of the objects of the fluid inside inside of that square. So if you do it for all of it, you can see that, for example, these 3 ones are, this area here, this square here, the more similarity is a little bit more going upright, and so indeed the similarity function find the maximum of similarity going upright. You do for the other, then you can estimate a velocity map, and here you can really see the velocity of your experiment, no? Because you have different, each arrows represent for you a movement in time, so a velocity. Now, this part is a little bit mathematic, but I hope it can help to understand better and to maybe correlate with stuff that you have done maybe when you do image analysis. So one very used generic measurement of similarity in not only in biology but generally in mathematics, in signal analysis, in physics is the cross correlation. Okay, so you can apply this cross correlation to everything you want to compare the similarity with. So you can go for a 2 series number, signals, images. What it is in mathematical terms is the convolution of the two things of the two objects you want to check for similarity. So it can be images, it can be signals, it can be whatever series of number. And then, for example, I don't know if you see this convolutional operator, maybe you have already seen in the filtering of image analysis, the same. If you have seen the convolution operator in the filtering of images in the other lectures, or when you study by yourself image analysis theory, it's really the same operator, the convolution operator. What it does is, at the end, is quite simple. It's comparing, it's just multiplying and summing index by index. So the correlation between A and B is like the sum of 1 multiplied by 2 plus 2 multiplied by 3 and so on. When you do it between A and B and A and C, the highest value you will find, in this case the correlation between A and B, it's where you have more similarity. So in this case you can say that the signal A and the signal B, the series number A and the series number B, are more similar than A and C, just because it's higher, just because of that. There are some problems with this kind of approach. One is like the magnitude of the signal can impact, the number of zeros you have can impact a lot. So they invented a normalized way to calculate this correlation, and this is called normalized correlation. The normalized correlation, it goes from between 1 and -1, and then so when it's 0 there is no correlation, and when it's 1 there is an high correlation. And if you apply the same thing, this normalized correlation to the signals we've seen before, to A and B, you will see that, for example, between A and B, you have a 0.9, so extremely high correlation, and instead between A and C, -0.1, so very low correlation. This is another example. I try to put many of them I hope to give you a better idea of what we are doing with our cell streaming experiment. Here, it's important to understand that you can use this kind of approach to find, for example, the distance, the lag between two signals. Imagine you have this signal A, so the orange one and the blue one. And then you want to find a way to say, okay, for how many time points from 10 to, like, here, there are time points, so if you move ahead of 3 or 2 or 1 time point, it will be more or less similar. So you want to really check when they will be more similar. And if you apply this approach, and then you calculate the normalized cross correlation, moving your signal a little bit ahead of one step by one step. Imagine to take the blue one and put it and move it ahead of 1 point. You will see that it starts to be quite similar to the orange one. And indeed, if you calculate the normalized cross correlation, you will find that there is a very high value when you calculate it with moving the signal blue of 1 time point. Then you see that the other ones are very low, except for example, -4. Why -4? It has also a very high number, because if you take this blue one and you move back of 4 time points, you bring, for example, this one on top of this orange one, and you find a high similarity. So it's important to not really to really understand everything about this one in 5 minutes, about this topic in 5 minutes. It's important to understand that there is a mathematical way to identify the similarity between signals, or between numbers, or between images. And then you also have the opportunity to use it to see the movement that you have to apply to a signal or to an image to be more similar. And then you can find, for example, in this case, that if you move of 1 time point, you will find a very high similarity, and this is an information that you can use, for example, in what we were searching before. The only thing is that in this example, we were just having 1 dimension, not only the time, like 1 dimension, going right and left. Here, you can see that you have a 2 direction, because an image is X and Y, so you have 2 dimensions, but the principle is really the same. So for each of these square, you can apply a similarity function. In our case, we are using normalized cross correlation. When you find the highest value in X and Y, it will be where you have the maximum similarity. And and what you are really saying is that if I move, I don't know, my image of 40 pixel on the right and I don't know 45 on the vertically up, it's where they are more similar. And then if you think what we are observing, what we are analyzing, that means that there is a movement of 40 pixels on the right and 60 pixels on the top. And this is like really a movement. And then since you have a difference of time, because you are having a time point, a time lapse, you have a difference of in space and a difference on time. It's a velocity. It's like microns per minute, microns per hour. And so this is you do for all of your square or your windows that you have decided to grid your image, and you have a velocity field. And from there you can really apply all the velocity field theory that you can find in the world, no? So for sure you can imagine that physics people will have 100 of theory about fluid and liquid movement in the world, no? So you can really, if you really want to go deep, you can really open a physics fluidity book, and you can start to check a lot of stuff about how your cells are moving in your different conditions, with a treatment, with a drug, with cells that are coming from a tumor cell line, cells that are not, that are sane, healthy. So you can really think about and then say, okay, it's a very useful tool now, because I'm able to use a whole field of the physics that is the fluid dynamic field and apply to my biological problem. Usually you do something more simple than all of those things. The first thing you want to extract is the velocity, okay? So you just have to imagine that you have, as I was saying before, this kind of analysis brings you for how long you have to move on the X and for how long you have to move on the vertical axis to have the similarity. So your arrow is really the composition of the movement on the right and the movement on the top, on the horizontal, on going up. So this is really a theorem of Pythagorean theorem, and to find the real magnitude of the velocity, so in combining the horizontal and the vertical component, you just really use the theorem of Pythagoras. So the square root of the square of the x component at the power 2 plus the y component to the power of 2. Then of course, and these are very important parameters, the basic parameter, if they go faster or slower, it tells you, no? It's the longest it is arrow, the fastest the cells are moving. Then you can of course extract some directionality the same way, no? This you can calculate the angle. So you can calculate if they are moving all in the same way, if there are more, how can I say, more variability in the direction, if they move all fast, but all of them, they go where they want, or if they move slow, but they move all of them in the same direction. And then there is something a little bit more complicated, and I really don't want to enter into the math details of it, but there are ways to calculate for how long the cells move in the same way. So you imagine you have a lot of cells in your field, and then you want to say, okay, in a condition, they have a similar movement for 500, around 1000 micrometers. In another condition, they have a coordination of movement of the same similar movement for, I don't know, 300 microns. So you can also extract more complicated parameter. So at the end of the day, what you observe, you do it for all of your frames, for all of your square you have drawn, and then you extract those arrows, and here under the arrows you will see a real experiment that you are trying to follow over time. So the arrow should tell you For example, here you can see that they are moving up, and so the arrows is following like this. In this area, they are moving. Now there is more coordination because they are more overgoing everybody up. And then this is how it works. This is analyzing a confined movement cell migration experiment. Okay, here of course you can do something more. This is like a real experiment we have performed with different treatment. What you can see is that there is a velocity increase in one condition, there is more correlation in one condition. Here you can see the angles, the orientation. You see here is more colored. It means that there are more different angles, more different directionality. In this one you can see that they move faster because the hours are longer, but also that there's more yellow, so the movement is more coordinated. This is the kind of stuff you can do with this kind of analysis. Okay, let me see. I will tell you also at the end, but it's important to notice that this kind of analysis, you can do it without any fluorescence marker. This kind of experiment are done with phase contrast, so with with bright field microscopy, wide field microscopy. So you don't need to tag your nuclei, you don't need to put any fluorescence in your system. You can use, or on the other side, you can use your fluorescence marker to mark other kind of structure you are more interested, but to do the motility assay to see how they move, you really don't need to tag them. And it's a very good thing. It's really useful, and is one of the reasons why is used this kind of approach, because it avoids you to do the fluorescence sample preparation, the fluorescence use, it avoids you to use one of your marker to do a marking of nuclei or something like that. You can use directly your experiment without any fluorescence. So it's rubber free, to be technical precise. Now, in the next hour, I think, 45 minutes, I hope, we will see how to analyze wound healing experiment. Wound healing experiment, as I was saying before, is quite similar, but you grow your cell in the same way you have grown to do the cell streaming, but then you scratch in the middle, and you want to see how fast they close the wound, and in particular, this is a directional experiment because you are imposing a preferred direction, no? Because the cells more likely want to go on the direction of the wound. Okay, starting from here, let me go out from this. We will try to do a little bit some hands-on, or I will try to involve you a little bit more but it's okay. Also, if you don't want to be too much involved, I will go by myself. But I just want to ask you, how will you approach, for example, this experiment? What you want to do? How can you think to estimate the speed of closure of this experiment with the tools and with the things you have learned in these days with Rensu? So what you think it can be a good parameter able to estimate the speed of closure of an experiment like this? If someone has some idea, just consider that I don't see the audience. Now I see the audience. Okay. Okay. If not, I can give by myself. Someone has some idea?
Yes. So you can extract those parameters with the measure function that I showed you. So what parameter might be useful?
How will you approach it? If you have some ideas, not, it's okay. Yeah?
Remember that we had area, perimeter, form factor, aspect ratio, all of those are parameters. So what would be helpful? Yeah, so we've got an answer here. Area would be helpful.
Yeah, the area, you can try to extract the area, no? So, and then, so this is a very good idea, and is usually how you can approach it. So you want to extract the area of the cell, for example, for the cell layers, we call them cell layers, and then see time after time how the area of the cell will proceed, will progress in our experiment. What we expect after doing something like that, we expect to end up with something like this. For each experiment we have, we end up, we can, I can say We can think to obtain something like that. No, each time point you have an area covered by the cells. So at the beginning you start from quite empty. Here there is the whole image size, so the full area covered, and then little by little you cover all of it, no? Because each time the cell layers progress and they cover a little bit more of the wound. Okay, so having something like that, how can we? So here, imagine you have to think about that. Here you have the micrometer square of the area, okay, and here the time. From a graph like that, which kind of thing we can do to extract the speed? We can think about to fit a line, no? We can fit a line here. What it means to fit a line. Find the best straight line that interpolate, that pass better on our raw data that they are the blue points, no? The blue dots. So why it works? Because if we are able to fit the line, we will obtain really like the one we do in math in our math, even high school, I think. So there is an intercept plus a slope multiplied by the time. The slope itself, it's a micrometer square per minute. So this is the real speed of closure, because it's the real, it tells you exactly each minute how many microns, how much area you are covering, how many micron square you are covering, you know? So this is like really good, no? If we are able to do this, it will be good, because for each experiment we will be able to extract the speed of closure. Okay, so now we will try to do a little bit hands-on. I will see how far we can go to do things together, and if it takes too long, I will go by myself. But we will start, I think 40 minutes, it will be okay for us. So you can open your Fiji if you have, I think. For me, it's taking one year. Okay, it has been open. And then you can open the S8 image. So S8 treated should be like that. Okay.
I'm just going to give people time, and then I'll tell you.
Yes, yes, you. Can you still see my things, the screen? Okay.
I can see everything.
Yeah, okay.
Everybody happy?
I suggest to open the S8 because we are using it in the PowerPoint, so we will be able to be everybody the same.
Sure. I think you can carry on.
Okay, so from here, what we have to do, as I don't know the name of that girl, but as she said, we want to find a way to identify the cell layers. So, like, first of all, as always, sorry, I always forget this, but when you start a new workflow, a new analysis, and you don't know, and you want to do test and trial, it's better to duplicate your image. So you don't need every time to reopen your image. So first thing maybe you can do is just duplicate the image so you can do some tests here. So like here you have to think what I've seen in these 2 days that can help me to identify the cell layers against the wound. So we want to find a way to segment these areas, and not this area, I mean, like segmented cell layers. So here, usually when you start with something like that, you can say, I don't know, I want to try some thresholding, no? If you see here, it's a little bit harder to find a thresholding directly, no? Because what's the problem is that if you see how it is the situation, I don't know if you see because the contrast is a little bit. Okay, now it's okay. You can see that inside the cells is quite gray, and then you have the borders, the membranes of the cell. So finding directly a cell thresholding like this, it will not be easy, and it will not work, let's be honest. So what you also usually do in some in this kind of analysis to think, how can I enhance, highlight my cell layers respect to the wound respect to the cell layers. You can use the filters, some filter, no, to enhance them. Do you have an idea? Anybody has an idea which kind of filter we can use here? You have to think what's the difference between this area, the wound area, which is the main difference between this main area and this area where there are the cells. Just think, I don't know, 1 minute, 30 seconds. Let's see if someone has some ideas.
So someone suggests a minimum filter.
Which with median?
Minimum.
Minimum. Maybe it works. Never tried. Let's see if it can be an idea. I think it's not a good one because you will create more disasters. Because you will really. But what you are doing with the minimum filter? No, you are going to in an area of 16 pixels. Let me see something, or 5 pixels. We are going to put the minimum. So you are going to really, how can I say, smash down, flat down everything to the minimum value of this that is very similar to the wound. So I think minimum is one that goes in the other direction. You will create an image that does all more or less the same minimum value that is very similar to the wound.
Someone suggests just the Gaussian blurring.
Gaussian blurring could work. It's a way. I don't. Maybe I never tried, but this one could some way work, but it will be still hard. So let's start to another question.
Just one last suggestion from the side. It was a maximum filter instead of the minimum. Try the maximum.
Let's see. That one can work really. This is, I think, one approach that can try to work. But let's do the real one after all the suggestions. So the real difference that you can see, maximum is close to a good answer, But the real difference that you see is that here there is more homogeneity. Instead, here there is more variation of the intensity of the pixels. So if you find a statistical mathematical parameter that explain for you the difference, the variation of something, of an intensities distribution, you will have a really good highlighter of wound against cell layers. And then it can be standard deviation, but in Fiji, the one is called variance. That is of course, is the square of the power of 2 of the standard deviation. No? Why it works? It works because it does something like that. You see the wound becomes really black because it's extremely homogeneous, so the variance is very small, and the cell layers will really be enhanced. If you put like 3 or 5, I don't remember exactly. Of course, if you exaggerate, you will obtain something extremely big, no? Because moreover, at the boundaries, you will find extremely variance, and so it will explode. But if you keep between 3 and 5, something like this, I think 3 is quite good. I don't remember which one I put in my, I think 5. Let me see which one I put in my experiment. Let's see. Yeah, I didn't put it. 1 second. Sorry. Okay, I put 5. So if everybody can put 5 also. And click OK. Of course, here, since you are working with a stack of images, you have to say process all the images, not only the one you are seeing. No, here you click yes. Let me see if I forgot something. Ah, no, I put this 5. I also put in the slides. Okay, so what you will obtain, and let's see if Renzo, if you can help me to see if most of the people, if everybody has obtained something like that.
So some people, when they duplicate it, they only duplicated the single frame. They didn't duplicate the stack, so they're just quickly sorting that out.
Yeah, this also, I mean, this experiment, this hands-on is also thought about, it's a quite easy analysis in a certain way, very basic filter and thresholding, but it will tell you how to deal with the image stack, how to deal, like I like it because it gives you a little hint of doing stuff also without a fluorescence marker, so you will see some little bit different stuff. Everybody's has put the filter.
Is everybody happy that the filter applied? Cool, yeah. Thank you, Ken. Carry on.
Okay. Now what you can do is at this point really go to the thresholding. Okay. And here you know you have different methods to try and see which one is good. Oh, here please put yourself on the first frame. Be sure to be on the first frame. Okay. So not be in the middle, just be on the real first frame of the experiment to try your thresholding methods. Here there are at least few of them that they are good. I forgot which one I used. I think I used Lee or Wang, maybe Wang. Okay, I think I used Wang. Something like that is quite okay, okay? Another thing about collective cell migration is like you don't really, in this kind of experiment, even if you have some little issue on your recognition, you are not trying to identify and find precisely everything. It's a more approximate way of study the migration. So you can, even if you have some something like this in the middle, or like you are not extremely precise to find to connect these cells here, it's still okay, because you will, at the end of the day, you will go to fit a line. No? So even if a frame is not perfectly recognized, it will still work your analysis. When you are happy with this, let me go back to my, okay, I think I put triangle at the end. Sorry, let's try with triangle to keep everybody on the same. Okay, triangle is better indeed. Okay, sorry. Okay, when you are fine, you click apply. Okay. Now you stop. You click apply and wait. Okay, wait. We have to do a discussion about it. So the first thing we have to discuss is if we want to convert only the current image or all the images in the stack. For sure, we want to convert all the images, no? Because it's a time frame, it's a time lapse, so we want to convert all of it. So here you put, you don't put the tick, you untick it. Okay. The first one, so, is a little bit more interesting to discuss about it. It's asking you if you want to calculate the threshold for each image. So what is it's asking you? First of all, we have to understand what's the question is, what the option is. It's asking you if it needs to calculate the triangle method frame by frame. So in the first frame, you will get out with a value of intensity, because you remember these one are automatic methods that extract the intensity value based on the histogram. Okay, so it will get the histogram of the first image. It calculates the triangle method. The triangle method says that everything above this number, 36, but above this intensity value, it will be considered of interest. It will be red. Here is asking you if for each time point, so I go to the next time point number 2, frame number 2, I see again the new histogram, and I will calculate again a value, no? So this value can change frame after frame because the histogram will be maybe a little bit different. Or this is if you tick it. So here you are telling to the image A, to Fiji, to calculate the threshold for each image. It means every time point, every frame, it will extract the new histogram and will try to find with the triangle method the new value. Or the other option, if you untick it, if you don't put the tick like this, you will calculate the intensity value of thresholding only using the first image. So it will find the histogram of the first image. It will use the method you selected, in our case triangle. It estimates the intensity value, and it will use, in this case, 6, the intensity value it has found on the first frame, for all the frames. So it will not every time calculate the new value. Which one for you? What I have to do? I have to tick it and so calculate the threshold for each image, or I will calculate the threshold and you on the first image and use it for all the frames. Just think a little bit about it.
Yeah, so the question is it better in this case to recalculate that optimal threshold value for every single frame, or would it be take the first frame as your reference, calculate one threshold value and apply that same threshold value to frames? Which one is better? Okay, so the general opinion here feels to be that it's you calculate it on the first frame and then apply it to all of the frames. You can share your opinion.
Stellenbosch is amazing. It's the first audience that reply good. All the other times they tell me to calculate threshold for each image. Instead, the good things to do is to calculate only for the first frame and then use it for all the frames. Why it works? Then you click OK and it will work for you. Please check that it has done something like this. Okay, so be sure. Oh no, sorry, here I didn't put the threshold, but be sure to be at this point. So like this. Then you click OK. Okay, you have to put this kind of option, and then you click OK. You will obtain something like that. Now there is the threshold. Let me remove the threshold. You should obtain something like that. Okay. Let's maybe it's everybody at that point. Is it okay?
Yeah, everybody's happy.
Okay. What's the point? The point is that for sure the histogram here, if I was no sorry, which what is it? The histogram on the first frame. Let me go to a frame. Sorry, people, because I clicked the wrong button. Let me extract the histogram. This is, for example. Sorry, I want to do it well. Let's wait a second. Let me apply the various filter here. Okay, so this is your first frame. If you see the histogram of the first frame, that is something like this, for sure is very different from an histogram at this frame, no? If you extract the histogram of this frame, you see it's quite different, no? So it will be extremely hard that the same method, in our case, the triangle method, will be good for an histogram like this, and then also for an histogram like this. This is because...
So can you maybe just click on the log button for both histograms? It might be easier to see the difference just that log.
Sorry, yeah, that's a good idea. Let me see if I can see also the other one. Yeah, it's... yeah, I don't know if you can see it well also. Yeah, I don't know.
You can see there's a little bit of a bump in the second one, so there is a difference.
Yeah, in the log way you can see the bump. In the black way you can see there is more. Here is very little distributed. It's all moreover all of them is here. Here is more like there is a little bit also a bump here, so you can see the bump. And so of course if you calculate the histogram for each time point, it will not work well. On the other side, if you keep the same value that you found on the first frame, it will be good for all the frames, because more or less all the variance filtered cell layers will have an intensity higher than 6. Okay, so it will work, and then instead the recognition is quite good. Now we are quite finished with this part. Now we measure stuff, no? Okay, let me see if I reach the point. Okay, so of course to measure you have to first of all select what you want to measure. In this case we are interested in the area, in the stack position, the time point, let's say, and the label. So it will tell you what is the name of your of your image. So be sure to put these ones, so area, stack position, and display label, in the set measurement. Set measurement is here, analyze set measurement. Myself here. Here there's analyze set measurement, and then you select the one area, stack position, and display label. Okay. After that, you can run the analyze particle. So first of all, we put what we put. We put a size filtering because we want to get rid of this object in the middle. No, so these ones are for sure other cell, debris, noise of your image. Okay, so the small ones we want to keep only the big ones. So I don't know, here you can put like 10,000 if I remember well. Let me see which one you put. Yeah, okay, you can put 10,000, and then you will remove those cells and debris and noise in the middle of the image. Okay. Then you want to display results. And then, do you want to exclude object on edges or not?
Yeah. So someone says, well, they're all on the edges, so you definitely don't want to exclude it.
Yeah. You don't want to exclude. You want to include only them. Yeah. Okay. Yeah. If there was include only edges, you would have used that. So here you are happy, and you say okay. Add to manager and display results. Okay. So nothing. In this case, we want to keep it like this. And of course, you have to tell them to process all the images. Okay. All the stack, the whole stack. Okay, so if everybody has reached up this point, so something like that, so the result here with the area and the ROI manager fulfilled with objects, it means we are good. We are moving ahead. So another good thing, another reason why you duplicate your images at the beginning is because now you can show and put the labels you found in your original image and see if the recognition is good. So let me know if everybody's here, able to see like this.
Wow.
If you don't see like this, you have to be sure that you set a setting. Let me see if I have on the. I don't have here. Let me see. Oh no, I have it here. So you go, if you don't see well like this, okay, it's because you don't have these settings. That is on more option. You go to ROI manager, you go to more option, and then you have to be associate show all with slices. Let me know if you have found it. If it's like on the ROI manager, you go on more and then you go on option.
Okay, Leonard, I think you can carry on. Most people seem to be sorted right now.
Okay, we carry on. Okay, so when you are, imagine that there is not me, and then you are alone, and then you have performing this experiment for the first time, you are analyzing it for the first time. What you do when you have such an experiment? First of all, you want to check if the segmentation, if the recognition is okay. So in our case, it seems very good. No? We are able to really find the boundaries of our cell layers. The second thing you check is how they are the results. No, let me go here. So the result is something like this. Okay, if you open your result table, okay, you will have something like this. Okay, Here, I mean, they are still okay. I'm not saying that they are not okay, but we have a little problem if we check them a little bit well. We have a problem that for each slice, we can have multiple areas, no? Because, for example, in the first time points, you will have the cell layer of the left and the cell layer of the right. But then when you go more when they are touching, you can have, for example, sometimes a single, you can start to have a single value per each time point, no? So, I mean, it's not a disaster, but what it will happen if you keep the table like this is that you will have some issue on the data analysis part, no? Because or if you are using Excel, Google Sheet, Python, whatever you are going to use, you will have to put in the data analysis part. If you want to plot, remember we want to plot our area over time plot, no? So how the cell layers are covering the image, the wound. So what you are going to do is to find some trick, some data analysis script to be able to handle the fact that sometimes you have two values for each time point, and then sometimes you have only one value, okay? So you can do many things in this case. In this case, you can put effort in data analysis with Excel, Python, R, MATLAB, what you use usually to do data analysis. And it's for sure a good way to approach the problem. But since we are in the image analysis course, we will try to redesign our workflow, so our image analysis workflow, to fix this. What we are trying to fix, to be clear, we want to have a single value that can help us to identify the area covered for each time point. So we want to be out, we want to find a way to have only one value for each time point. Okay.
Sorry, I just quickly want to say something too. So just to be clear, what happened here is when you click Analyze Particles, it found the two sides as two separate particles. So there are two areas at every time point now. And once they join, as you were saying, then they're now joined to one particle, basically. Yeah, that's something. Yeah, so just to say, it seems like also an issue that's coming up for at least some people is that when the areas are extracted the labels, you know, of where it's found the segmentation, it just shows all of those labels on the every single slide. It's not showing
that one we can fix it soon. I think quite soon, because there is also a slide about it, and this year to fix it, you have to go to ROI Manager, go on More and Option. It will show you this. More, you go to your ROI Manager, you go to More, you go to Options, and then you do Associate Show All ROI with Slices. That means that now it knows the position of the ROIs, of the cell layers. After that, if you go and click Show All, it will be adjusted. It will be fixed. So you have, if you want to fix it, you go to More, Option, and then Associate Show All with Slices.
Yeah, thanks. That did fix it. Thanks, Daniel.
Yep. Okay. Okay. What we are going to do? Some problem?
Yeah, everything's right now.
Okay. So of course, to move on. Okay. There will be 100 ways to fix this problem. I'm telling you, to have a single value for each frame, for each time point. We will do one of them, okay? But the important thing when you redesign a workflow, like this thing we are doing now, so we have a workflow that was working quite well, but not extremely well, not perfectly. When you redesign your workflow, you have to think what is good and what was not going well. So in our case, the segmentation was good, no? So the segmentation was good. We keep it, okay? That one was perfect. Our cell layers were perfectly identified. So the problem is not there, okay? But we want to work a little bit on find a way to obtain a single value. This one I don't want to do like for real hands-on, because I honestly think it's quite hard to find a way that you can think about it. Not because you are not smart enough, because it's a little bit complicated, like it's a little bit complicated, and so I prefer to do it by myself and you just follow, okay? So you close everything. I close everything because I just want to keep my where I reached. So here, let's see this one. Let me open again the image. Then I have my mask that I obtained before. This is good, but what I want to do is to do some step before the calculation of the results that allows me to get out with only one value. So the first thing I do is do an analyze particle, and instead of directly display the results and do all the analysis, I extract the mask. What it means extracting the mask? Extracting the mask means that as a result, I want the filtered mask. Filtered in the sense that I'm filtering by area. So I'm removing all the object with an area lower than 10,000. This helps me because I need to do a step, a pre-processing step before calculating what I'm interested in. I say always okay. If it works well, it will show you something like that. This one, I need to like invert my root because I want to see white and black and white. Let me find it. Where is it? Okay, something like that. Here you can also try to don't follow me too much in this. Like I mean, not to follow, not doing it with your Fiji. We will do it step by. I will do it step by step. From here, I can decide to extract as a measurement. Let me see if I have here. Yeah, I'm already reached this point. So we can extract the so-called area covered percentage. Let me see how it's called in Fiji. Where is it? Area fraction. So the area fraction, and the area fraction is the percentage of area with a white pixel respect to the whole image. Okay, so for each time point, it will tell me here I have 20%, here I have 50%, here at the end of the day, here I have more or less 100%, no, because everything is white. To do it, you can do in different way. I will just show you one of it. Let me see where I put it. Okay, here. In the presentation, we'll find everything. What you have to do is to select all the image with, where is it? It's select all. So you want to do a selection of the whole image. Then you add to your ROI manager, because you want a measure for the whole image. So you need to tell to the program that you want to analyze the whole image. One way to do it is select all and then create a ROI, a region of interest, that is the whole thing. Okay, I put it here, and from here you can, in this very small, very hidden menu that is more, you have many interesting stuff. One is the so-called multi-measure. Multi-measure, it means it does, for the same ROI, for the same selection, it will perform multiple measurements for each, in this case, for each time point. So it's asking you if you want to measure all the slices and so on. Okay, let me see if I put everything or if I forgot something. Let's show. No, I forgot to put, sorry, I have to redo because I forgot to put the measurement. That is this one, okay. Let me do again. Okay, so now what you will see is that you have the area of the image and then the percentage for each time point. So for us, then it will be easy to find the area covered, just multiply the percentage of area covered for the total area of our image. And this will give you what we wanted. So a single value for each time point. Okay, so now this is what you will obtain. You will have Excel file in the folder I put yesterday. And now we can perform the so-called data analysis. We will try to do it all together. It usually takes 20 minutes, 25. If it's becoming too late, I will push it faster, okay? So what you have to do is go into your, where you have your Excel files. I don't know if you want to use Google Sheet or Excel. I think on the presentation I used the Google Sheet, but it's really, we are doing quite basic stuff, so it will work for both Google Sheet and Excel. And then if you open this file called area over time row, you should have something like that. If you have many other stuff, you cancel everything, because I forgot to cancel. I put it with already the calculation, but we want to do the calculation together. So you have to open your file and have something like this. If someone else has something like this, let's show you. Something like this. With all the calculation, everything already done, you cancel everything and we will do it together. So you should have only file, area total, and area percentage. Or like label, area total, area percentage. Let me know if everybody has opened this one. Everybody has the Excel file open?
Yeah, seems like it.
Okay, so now we will try to do step by step and see how it goes. So the first thing you need is to calculate the area covered, no? And the area covered, I mean, it's really what we have already said, is really doing, let me show you what you want to do. You really want to do the area total multiplied by the percentage. Remember that this percentage is already multiplied by 100, so you have to divide also by 100, okay? So you should obtain something like this. Let me put it here.
Sorry, Emmanuel, just wait a second.
Okay. From Lisa, I cannot hear.
Lisa just said that everybody must just delete their graphs first, delete the extra fields.
Yeah, that's
so they can get to the
yeah,
same
it was my error because I put the one already analyzed. So you should really start from something like this. Here you should have you can write what you want, of course, as a title of the table, of the column. Then you here is important to do B2 multiplied by C2 divided by 100. And then autofill. This one is really helping too much. And do the autofill, okay? So the formula is this one. Where is it? I lost the formula. Okay, it's here. The formula is this one. And then you drag and drop, you know, double clicking you, it will be auto-fulfilling for the whole column. Okay, proceed.
Okay, yeah,
the second thing, very important in biology, is that you are working with real physical objects, no? So you can calibrate your measurement. In this case, for example, this experiment was taken with a time frame of 10 minutes. So each time point is 10 minutes. And then a pixel is 1.6. So you can calibrate your, you must calibrate your system. The first thing you have to do is to create one column called time in minutes. Then, of course, the name of the column is completely up to you. Then you can put 0 and 10. And then, as we have done before, we can auto-fulfill everything with the time points. And then here, we can also do the same for the area covered to convert it to physical units, in this case micrometer square. Here, usually what you do is to multiply the pixel you have by the physical unit of your camera, of your calibration, to the power of 2, okay? and then you do the autofill. For you, you put a dot and not a comma, eh? Because for me as Italian, they like to put comma, but for English people, is a dot, like it's this one.
I think you can proceed.
Okay. 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 From here, what you can do is finally plot your. I think it's what we can do is plot our area over time. No. So I don't remember in Excel where is it, but here the point is that you have to select the two column, and then from here I also don't remember exactly where it is. From here is a chart, I think. And then you do all line chart or scatter, the one you prefer. It's really similar, but I think we can do the scatter plot. I think it's better. Let me see which one I picked on. Yeah, you should find a scatter chart somewhere. Maybe like this. Okay. Okay, something like this. You should obtain something like that. So it's like a scatter chart, and then if you suggest you different stuff, you pick the one, of course, showing like this and not with two lines. Also in Excel it should be scatter plot. Let me see, I have it here. Let me see which one it is. But here is in Italiano. Sorry. Okay. So let me know if you have reached something like this. It's not really important that you do, everybody do everything. It's just to follow at least what we are doing. Of course, if you can follow everything is better. If you can write everything is even better, but everything is on the slide. Everything is, it's more important to understand what's going on.
Yeah, I think you can carry on.
Okay, so from here, this is perfect, no? It is exactly what we wanted, of course. This is very good because not always it's like that. Not always what you think you will obtain from your experiment is what you get. But in this case, we really did a very theoretical good study, and we were quite sure that the cell area progression would have ended up something like that. From here, let me see. So from here, we really want to extract this fitting, no? Of course, in Excel, in Google Sheet, there is you can do that, no? There is the trend line. There are a way to do this. But what is our problem, if you think about it? What's our real problem? If you see a problem, if we go directly like this and ask, for example, to the Excel to extract the slope from this data, no? From this data, you ask to Excel to obtain the fitting line and then give us the slope. What will be the problem? If someone has some idea about which problem we can get if we directly fit a line using this data.
Yeah, you say that.
The fact that it's flattened, so you're asking only the increasing plot.
If I understood well, you were saying that you only need a piece of the data, no? Only the beginning. Because if not, you will fit also these data points. This is the problem. The problem is this one. If you take all these points, of course, your fitting line will be something like this, no? Will be, I don't know, let me show an example. Inserisci, where is it? You will probably obtain something like this, no? Because it will also try to fit this point, no? So you will obtain something like this. So we have to find a way to get rid of this knee and of this plateau. That is when it reached when they, in this area here is where the cell layers starting to touch each other. And then here is when they really close the wound. And so we are no more interested. There's no more a wound healing, no? To that, of course, what we can really do is to find a way to get rid of this, to keep and analyze only the first part of the graph. One way to do it is, for example, to say, let's keep the frames up to a certain area percentage. So, for example, 80%. And we will try to do it in Excel, okay? How can we do it? we can, let's go back to Excel, to Google Sheet a little. Okay, here it's a little bit too big. Let me put this one very far away. So first of all, we have to create two columns that will be like time thresholded time points. So the one we want to keep, area covered thresholded. So thresholded in this moment will just mean up to when we reach the 80%. Then we can put here like a parameter that is the area percentage, and we can put in this a value that we want. So we put 80, for example, because 80 is a good way. You know, it's quite at the end, but not so much at the end. It's just picked a little bit randomly, but with a certain thinking. No, so it's not, you are not keeping a few points, but you are not already touched at the end, at the real end of the experiment. So this is the start. Okay, so you create space to have the time points up to when there is the 80% of the area covered, and then you create a column to have the space to put the values up to when there is the only 80% of the area percentage covered. Okay, so now it's a little bit more complicated, the formula, but it's still, I think, understandable. Let me go back to the presentation. What you can use is an if, no? So what you want to do is to put an if that says, if the area threshold is below my value, and this is also useful because we already extracted the area percentage, no? We extracted it from the Fiji algorithm, from the image analysis workflow. So if we are under the 80% of the area covered, we keep the time. If not, we put blank. And to put blank, to put empty, in in Excel and in here, you put this kind of double apexes, I think is in English. This, I leave it for you for a while, and you do the same also for the area covered. The only difference is that you, in the time point, you keep E2. In the area covered, you keep F2 because you want to keep the area, and then E2 is the time, F2 is the area. So you have to just pay attention, but the other part is really the same, okay? So now you can select all these points, you can select them, and then you can do the same things you have done before. So insert chart, go for scatter. I hope it works because I have some issues sometimes with them, but this is like scatter. Okay, you should reach a point like this, okay, with only this one. And now it's okay, no? Now it's fine because we have reached our only the we removed the plateau area, the knee area, and we only have the linear behavior we expected. Okay, I try to increase a little bit the speed because I want to finish before the 2 hours. But I mean, I hope most of the people have reached here. If not, I can wait 1 minute.
Yeah, I think everybody's happy. Okay.
From here, what you can do is to put the trend line. Let me see if I find. In Excel is somewhere, in Google Sheet is here, but it's always called the trend line, okay? If you put your trend line, you can have a different option. For us is a linear behavior, and then we want to show the equation. Maybe we can put in red to see it better. Sorry, I just want that at least you can see it. Okay. Okay, so you should have obtained something like that. Let me go back to the PowerPoint that things are a little bit bigger. You see? Oh. You should have obtained something like this.
Everybody's got the formula. Yeah.
You have to add the trend line and then you label with equation, and it will put for you. Okay? I hope it's. And so this is we are finished. I mean, if our scope, our purpose was to find how many micrometers, how much area in micrometers square the cell were moving, this is really the speed of closure. So each minutes they will fill 537 more or less micrometers square. Okay, so this is the real speed of this experiment. Of course, in Excel, you can, in whatever, in Google Sheet, you can have also the formula to calculate it. And in this case, instead of putting the line, no, you want to obtain only the number, you can use slope, and then you have to be sure to get the area covered. As first column, you want the area covered, and as second argument, it wants the time point. If you do that, sorry, let me go back here to the Excel. You should obtain the same number, okay? Well, if you do slope and then you should obtain exactly the same number that you have found when you did the thing with the trend line graphically. Okay, so the formula, I put it again for the last time, I think. Here, slope. And then it will show you the same value you have obtained in this place. Okay, and then it's over. We finished. Okay. Okay. So now I just want to show you in the last 10 minutes how very briefly why you can use Fiji to automate your analysis. It's not about that, the course, so it will be the next African Bioimaging Consortium community course, but I just want to introduce the argument. One thing you can do with Fiji is to record the things, the operation, the commands that you are doing. It's quite easy. You just have your macro recorder. It's like a button. You open it, and then everything you are doing, it will be recorded. So, for example, in our case, if you remember, we started with duplicate our image. We applied the variance filter, and so the duplication is recorded as duplicate. The variance filter is recorded with run, variance, radius. We put 5, so it also take the argument and the radius we put. It says that it was analyzed the whole stack, and then you can do it for all the steps we have done. No, so what you will obtain, you can create your script, you will obtain something like that, no? So we do the duplication, we apply the variance, we convert to a mask, we analyze the particles, we did the multi-measure, and so on, no? This macro you will find in the folder that Renzo shared with you yesterday. And then this is really a script. Then you can use it to analyze any other experiment, no, that you have, more or less. I mean, it's the starting point to analyze it. So let me just show you in 1 second what it means. I have my macro or script. Everybody knows, call it in a different way. I use it sometimes macro, sometimes script. So I have my Fiji. One here to open. I recorded it, I fixed it, and now I can really open another experiment, for example, the S2 that we have in the folder. And then if everything works well, I can just run it. So I go run. And ta-da! It has... It has extracted our data. No? So what I can do, for example, I can, I don't know, take this value. Let me try to do it fast. I didn't try it before, but it should work. I go to my Google Sheet here. Where is it? I can copy and paste my value, and everything will work, no? Because I also have a template for it, no? So I can just really do something. Let's do it in Excel, but it's really the same. I copy. I have to be sure that the column is everything organized the same way. So I bring the same name of the stuff. I copy them. Let's see if it works. I didn't try it on, but it should. Let me see if I can copy. You can copy all these formulas, and then you copy your new data, and it should work. Now we see if it's true or not. Let's see if it works, so I have to do something more. Let me see if it has worked out. Yeah. So now you have that, and then you can also take this one, the slope. And then what you see is that, for example, I don't know if you see because it's small, but let's enlarge it. The slope of the control of this second experiment is 135, and the slope of the treated, the one you helped me to analyze, is 536. So this meaning that the treatment increase a lot the speed of the cell, because the control one have 135, the treated one have 536. Then this is already an indication. No? They move faster in a situation, in a condition respect to the another one. And as you can see, this is just to show you how you can start to automate your analysis. Of course, it's not the course topic. I just wanted to show you that you can do this kind of stuff. Okay. In the folder you will find some macro to analyze them. And then what you can do also already with fish is to combine them. Like, let's do it. It takes 1 second. So I obtain my result, and then I want to check qualitatively by eye if it makes sense. No? So I can, for example, combine them. I put the control on top and the treatment on the bottom part. And then we saw that the control should be a little bit slower, much slower, I mean, because it's 4 times slower. Let me put a little bit of contrast. And then, so on top you have the control, so it should be indeed it seems much faster, the one on the bottom part, that go well with our results by analysis. So this is always useful when you start to analyze your data. It's a new experiment to have also a qualitative check. If you can do it, it's not always doable, but when you can do, it's also nice to do it, no? To have a way to check also by eye if the results make sense, or on the other way, before starting a very complicated analysis, check by eye if it's possible, if there is something that it's interesting to analyze better. Okay, so I think we are quite done. So if people want to think about how can you make this analysis more complicated, you can check if the linear fitting is good. In our case, it's good, but sometimes it's not so good. Linear fitting means constant velocity. So it means we are thinking that there is no acceleration, there is no stopping, there is no braking. The cell will always go to the same velocity up to when they start to touch each other. Then sometimes you are maybe interested if one side is faster than the other one. You want to maybe sometimes check if the cell layers move all together, if there is a straight line, a straight cell layer border, or they do fingers, invagination, or they move all together. You can try, but it's much harder, and I think we have done already enough today. So what we have learned, in my opinion, so I think one thing we have to think, it's important, even not so complicated analysis. Wounding, as you can see, is not such a big deal. It's really one filter, one segmentation, and fitting a line. So it's quite basic stuff. But even in something so basic, sorry, here there is an error. This is another course that you cannot. It's in Italy. It doesn't. It's an Italian course. So it could be tricky, no? In our case, for example, the first one was quite okay, but then we needed to rethink a little bit about it. Then it's very important to always have in mind what is the goal of our experiment. This is true for all the biological experiment, but here, for example, we really needed to have we have really clear that we wanted the area over time. So our goal was very, very clear, okay? And then image analysis is strictly related to data analysis. And for the more biological purpose in the room, it's extremely related to image acquisition and sample preparation. If you prepare badly your sample, if you acquire it wrongly, it will be much harder to do image analysis and data analysis. As you can see, the imaging macro language, the Fiji, the Excel could be a bottleneck. So this is why it's useful to learn real programming languages, Python, also for doing data analysis. But this will be another course for sure. But it's important to see also that even if it's harder, in situations like the one of wound healing, even Excel can be enough. It's just a little bit harder to deal with everything, do the columns, the things, the stuff, but it can be enough. But if you, as soon as you move a little bit more in complicated data analysis, you start to need more complicated programming languages. Okay, just this, for example, the paper we were talking about at the beginning, we have done with my group. Here you can see this is really what we have seen today. What we saw today. This is a wound healing analysis. So here there is the area coverage over time for two conditions. What you see is that here the behavior is not completely linear. For example, you can see more in the blue line, in the control line, that it seems like it goes a little bit slower and then it increases his speed, no? So, for example, we find a way to identify this point, and then we combine it with PIV analysis. So this is the angular, and let's say the one we have seen at the beginning. So using the arrows, the PIV analysis that we saw at the beginning, we can put inside a wound healing assay to, for example, extract the directionality of the angles of the movement, no? As we have done at the beginning with or in the theory part of PIV. And then, for example, here you can see that the lag time is a little bit correlated not only with the velocity, but also with the different correlation in terms of angular of angles, no, of directionality. Here, after 150 minutes, the speed, the fastest condition is already going all of it on the same with the same direction. Instead, the control not only is going slower, but it's also not yet organized. Instead, when they are starting around 40 minutes, you are quite sure that they have already reached another speed. They are more organized also, the control. This is just to show you that you can combine different kind of analysis. Okay, this one we saw. Another things you can do is to combine, for example, wound healing with, so you can find the wound. It's not drawn, but you imagine you can. It is also wound healing, but where we put the nuclei tracking, so you can track the nuclei, and then you extract, for example, directionality based on if they are close to the front of the wound, if they are not, they are in the middle of the wound, of the cells, or they are far from the wound. No? You can combine stuff. Of course, it becomes more harder, but it's also more interesting. Then, of course, you can go in 3D, and here is when you see this condition, you will see that move faster also in a 3D environment. Okay, conclusion. This analysis we have seen, it's really interesting because they show you that sometimes, not always, but not in many cases, but there are times where you don't need cell or nuclei segmentation. You can do an analysis without go to find the boundaries of each single cells. In particular, for wound healing, at least we had to segment the wounds, the cell layers. But in PIV, you don't segment anything at all. You just take the whole image as it is. It can go from simple parameters to like speed and to more refine, like velocity field description, like coordination movement. It's only transmitter. You can use it without fluorescence. You can extract this parameter just with your image, without label free, just with the transmitter light. But if you want, you can combine it with fluorescence and do like this kind of analysis we showed before, like this one. Okay, I think it's done. Thank you, everybody. If you have any question, everything, just ask me or write to Emanuele Martini. And for sure, follow African Bioimaging. That is very nice.